Hello everyone. So this is part 2 of exploration. We are going to cover points from 10 to 19. We covered from 1 to 9 in the last video. So the point 10th is equipment is listed with size 100 ml 100 ml beaker, 200 ml measuring cylinder, whatever you use, the number, how many, and the uncertainty. So these three things should be listed with clarity. So if you go and check the samples provided, now this sample where you have the things there in the heading under materials, it's not neither apparatus or equipment or chemicals, is just material. So simply it is covering everything. Another sample is this one, which is provided from one of our students. So you can see that this one is much more clearer and detailed, and this is by giving them instructions that please provide detailed information. So you can give the heading all three together, but when you are doing the material, please make sure you're putting material only. You are not putting apparatus there. Equipment, apparatus, only equipment and apparatus, and whatever chemical used you can give separate for that as well, because here you are for chemicals and material, you are not giving uncertainty. Uncertainty is only with the equipment or apparatus used, and that one is the smallest measurable unit. So for example, if you have used the stopwatch, other you can see the smallest measurable unit for measuring cylinder 0.5 or 0.1, for 10 cm cube is 0.2. Electronic weighing balance can be 0.1 or 0.01 depending on the precision of weighing scale you are using. The stopwatch, the same, it can be 0.1 second, 0.01 second. Now here, if you are including the human reaction time, please put it in the bracket or included human reaction time. It's better to provide the clear information if the precision is 0. Smallest measurable unit is 0.1, and if human reaction time is taking, it becomes 0.5 second, and it is unit given, and the units are SI unit. It is not given in ml, as some of you have made the mistake. It is cm cube, SI units are given. So make sure these details are correct, and double check with your procedure that you are not missing anything here in the list which is you are using in the procedure. So these procedure and this list must not be contradictory at any point of time. So this was point 10, which we're talking about. Method to collect data, address research question. Now in research question, if you have mentioned that your collection of data is using the spectrophotometer, or it is with the like this research question is not very clear, because I'm not I would say this research question is not that detailed, because I'm not getting the idea how the raw data is getting collected here. But if you look at this research question, you can see the spectrophotometry is used to collect the data. If I'm using a spectrophotometer, it means my procedure should clearly indicating that how data is collected. And that is the point 11. Method to collect data is clearly addressed, which is in the RQ as well. Method is presented in a stepwise format and can be repeated by others. I do remember some of you asked that paragraph. It is already written in the checklist stepwise format, so that is easy to repeat. So if you want to make sure that your procedure is perfect, you can ask some other person to read just the procedure and ask them if it makes sense to them. If this procedure is given to them, can they follow the steps clearly without any ambiguity, without any confusion? If it is the case, it means your procedure is clear and simple. So here it's a little more in the form of a bit it's given in very few steps, not very clear, but if you look at this one, the steps are very, very clear, broken down into numbers. Sentences are not too long, easy to make sense, and some of you have given the clarity of all the steps, that why you are doing so it's very clear. So in this one, and something which is, few things are simple steps, like you're cutting and then you're using a blender or a pestle mortar. Measuring cylinder is to measure this and that, but few things like it go more in detail of chemicals, then probably you can give more details about that thing. Here the references is given. I prefer to have referencing of the methodology. So you can do in the beginning, before you give me the procedure, little introduction of the procedure, or you can give a heading methodology and give some intro of your procedure along with referencing of your procedure from where you have adapted and what changes you have made. So as we are telling you, we are looking at the quality work here. So to make it a quality work, I need to make sure that it is clear, and referencing is given. This clarity, you can give the methodology little bit. Few can, what I said can, it means to make a quality report. It is not compulsory that if it is not there you're going to be penalized. It's just the marking for the overall report. Point 13, method of manipulating IV, minimum 5 over a suitable range. So we have the independent variable and it should be given in the suitable range. Now your independent variable can be 2 or 3 as well, depending on your investigation. So I would little bit go more about 13, the independent variable. If you are looking at the association of two variables, and you have of two variables, like this variable is affecting this or not, so your independent variable comes here, your dependent variable comes here, and generally it is a line graph with 5 variables. But if you are comparing the variables itself, apart from looking at the association, your dependent variable, whether this is increasing the higher, and then I'm comparing these two. So for example, I look at the concentration of juice, of some specific juice, and this is the concentration of juice is affecting the vitamin C, the concentration of vitamin C present in the juice or anything. Then it's like I'm looking at one particular juice, and in this juice I'm taking 5 different concentration and different concentration of vitamin C by doing DCPIB or any other method of doing that. But if I want to see that how much vitamin C is present on juice A, juice B, juice C, juice D, and then I want to compare whether this one is higher to this one is higher to this one, or this one to this one, or this one to this one, or this one to this one. So it's like kind of extension of my investigation. Then I'm doing the comparison of two variables as well. So if I'm doing the comparison of two variables, it means it requires for me to do the t-test. For t-test, 10 trials must minimum. It can be more than 10, but minimum 10. So that's why it is written that method of manipulating IB is given. How you're changing, for example, if your independent variable is concentration, how you're changing the concentration. If your independent variable is the type, how you're changing the type. Let me go back to the research question. So this one is having three brands of, so these are how, so these are brands are given. If the concentration, how you're preparing the concentration should be clearly given. A banana packing, risk packing, and control environment, I would prefer with this with more clarity, with more detail. It's this one, right? So this is what we are talking about. Sufficient repeats, as I told you, 5, definitely we need 5. There's no minimum 3, it needs to be 5. But if we are doing the t-test, minimum 5 for a standard deviation, 5 repeats is a must for S standard deviation. So it means because we are doing a standard deviation with all the calculation, minimum 5 repeats, but minimum 10 repeats for the t-test. So we are not looking at 3, it should be 5 for reliability. We are looking whether the data is reliable by doing the standard deviation. Method to measure the DV is given in the steps. It should be very clearly mentioned in your method that if you are calculating the rate, what did you do if you are calculating the counting the number of bubbles and looking at the rate. So all these things should be mentioned in your procedure clearly. Annotated photograph, you have given the photograph, the setup of your experimental setup, student experimental setup, right? And it should be referenced, the picture should be referenced as by the author or by the student. Name should not be mentioned there, but it should be referenced. Photo should be annotated clearly that what setup you are having. Reasons for how results will be presented, raw data transformation, statistical test are given. So if you see in this sample procedure, I don't exactly remember, but few, yeah. So tabulate the results. This point here, tabulate the results, calculate the average, obtain the nitrate value. So this is step. If you are whatever a statistical test you are using, you should mention that in your procedure as well. Safety, ethical, environmental concern, address including animal policy. If some of you have used, for example, meat or meat products or prawn or any animal product, you have to mention in ethical concerns about the IB animal policy. It is there in the Firefly and the manage bag and the resources provided to you. So, and you must know about the IB animal policy, so please mention as ethical concern there. You, if you have used food, so you should talk about the ethical and environmental concerns there. This should not look superficial. Please think critically. As an IB student, we want you to be more open-minded. We want you to be more critical thinker. So you should able to connect things by now, that if I'm using seeds, though it is just handful of seed, but still it is a food for someone. So how to make sure we are not wasting it, and if you have used it, after use what you can do it, how you can recycle it appropriately. So all this comes with a proper critical thinking. safety concern, whatever. If you're talking about your chemical which is harmful, that is not your information, and how harmful they are. So for example, in this case, if it is the student is saying is causing skin irritation, how do you know that? It's suggested to, you can give a table form as well, where you are giving the chemical picture and the safety precautions along with that chemical used. General safety precaution with your water bath, temperature, your safety precaution with your glassware, all these comes in the safety precaution. But this part gives a very clear idea that how well the IB student can think of different aspect, not only scientific but different aspects as well. Full citation is a must. As I said earlier, that your procedure methodology used must be referenced, and from that referencing it should be clear that how well you are able to adapt it. Means definitely it is just the you're getting a kind of idea from that. You're not supposed to it's just a type of referencing that you read, you understand, you adapt it in a way that suits best to your condition. So all this should be included, indicates with your full citation. And definitely if your exploration is very very simple, and it is a kind of exploration which is very common, and there's no these parts are strong critical thinking about this one, you are not able to give details of your things, the procedure is very vague, uh, so the high chances your exploration will fall below 3. But with clarity, simple steps, simple investigation, but with very very good clarity, though it is a very common investigation, still you can gain around 4. But unless it is a very good and clear quality exploration. I guess that's all. You can have a look on the sample. Sample 2 is a very high quality sample. You can have look on sample sample 1 is a high quality sample. Sample 2 you can have a look to get the idea that how, if it is not very detailed, how it looks like. All right.